This video will focus on a summary of all of the videos thus far. What we've covered in the last 4 videos is outlined here on this slide. Initially, we discussed the structure of skeletal muscle cells and talked about the motor unit as a motor neuron innervates individual muscle cells and has a neuromuscular junction, the critical components of which are highlighted in this graph. The motor neuron axon depolarizes, calcium channels within the membrane open, causing vesicles within the terminal axon to fuse and release acetylcholine into the synaptic space. The acetylcholine then binds to acetylcholine receptors on the motor end plate, which activates sodium channels and generates a membrane depolarization, which is transmitted via T-tubules and a subsequent flux of calcium out from the sarcoplasmic reticulum into the interstitium so it can interact with contractile proteins via rhinitidine receptors. These contractile proteins include thick and thin filaments, thick filaments being predominantly myosin and thin filaments being predominantly actin. When calcium fluxes into the cell, it causes actin to bind to myosin, and then you get the release of this phosphate and adenosine diphosphate, which causes a conformational change within the myosin fiber that corresponds to shortening of the sarcomere. ATP then can reattach to the myosin head, becomes hydrolyzed, and the muscle fiber is once again, and contractile proteins are once again in the resting but ready position when a subsequent calcium activation occurs. And picture 5 here gives you more of a schemata of what happens to the relationships of myosin to actin during this process. So this slide here, I think, nicely summarizes all the aspects of muscle contraction of skeletal muscle that we've discussed in the prior videos. I would like to finish up the video talking about comparisons between the various different muscle groups based on certain properties. This table summarizes the key aspects and differences between skeletal muscle, cardiac muscle, and smooth muscle. The top row is in reference to initiation of muscle contraction. And for example, in skeletal muscle, an action potential propagates down a motor neuron to the neuromuscular junction. However, in cardiac muscle, there's no motor neuron intervention, and the action potential is generated at nodal tissue, specifically the SA node and the AV node. And as I promised earlier, you will hear lots more about this in the cardiovascular section of this course. This could be contrasted to smooth muscle, where neurotransmitters via nerve fibers activate the muscle, but there's really no motor end plate. Varicosities in the nerve endings contain vesicles of neurotransmitters, both sympathetic and parasympathetic. And action potentials and neurotransmitters are not the only mechanism for muscle activation. Also, stretch, O2 tension, and pH changes can precipitate contraction. And if you think about it, smooth muscle has a very different role within the body compared to skeletal and cardiac muscle. And as a consequence, its activation is also quite different. Now, sarcomeres exist in skeletal and cardiac muscle, but not in smooth muscle, which is why it's non-striated. The innervation for skeletal muscle is somatic only, whereas autonomic is present in both cardiac and smooth. And excitation contraction coupling in skeletal muscle, like we spent quite a bit of time talking about, is not calcium mediated, but rather results from an unplugging of the ryanodine receptors on the sarcoplasmic reticulum, which then leads to calcium release. And as a consequence, extracellular calcium concentration does not impact strength of contraction. This, however, is quite different to cardiac muscle, where it is calcium mediated, where there is calcium mediated calcium release, and that the action potentials result from depolarization of T-tubules, inward calcium current, which then activates the ryanodine receptors to release further calcium from the sarcoplasmic reticulum. And in this circumstance, extracellular calcium does impact on the strength of muscle contraction. Now, in the case of smooth muscle, calcium arises predominantly from the extracellular space, as the sarcoplasmic reticulum in smooth muscle contains much less calcium than it does in the case of skeletal and cardiac muscle. This muscle type does not have T-tubules, but rather calveolae, and extracellular calcium, similar to cardiac muscle, but different than skeletal, does impact strength of contraction. Now, in the case of skeletal muscle, strength of contraction can be increased by summation and recruitment, but there is no effect of epinephrine or norepinephrine on this muscle type, as it only receives somatic innervation. Cardiac muscle is dependent on extracellular calcium, similar to smooth muscle in that way, such that more calcium will translate into greater strength of contraction. However, there is no risk of tetany in cardiac muscle because of the refractory period that the muscle has to recurrent activation, which again is a very good thing, as tetany in a cardiac muscle would not be a physiologic advantageous experience. Finally, cross-bridge cycle. In the case of skeletal and cardiac muscle, calcium binds to troponin C, which results in interaction of actin and myosin and conformational head changes in the myosin, so-called the power stroke, which is supported from an energy perspective by ATP, which is pre-hydrolyzed to ADP plus phosphate, so that it is ready to contract as soon as a calcium signal is experienced. Now, smooth muscle is similar to cardiac and skeletal muscle, except it has this myosin light chain kinase, which must be phosphorylated before it can precipitate muscle contraction. And there is no troponin in that circumstance, but rather calcium binds to calmodulin, which in turn phosphorylates myosin light chain kinase. So that is an overall summary of many of the concepts that we talked about, and also compares and contrasts what I think are the key differences between skeletal, cardiac, and smooth muscle.